research guide in the field of zoo. Uh, 4 students are doing PhD under his guidance. He published 17 research articles in national, international level journals, successfully participated more than 20 national and international conferences and presented papers. Authorship of 9 books is also credited in his account. He also successfully completed 2 research projects, received funding about 2.75 from the UGC and STRIDE UGC. Before we move forward for our first lecture, I think I would like to request our principal sir to give his presidential address first. Then we will move to our first lecture. Thank you, sir.
Okay, good morning to all. We are gathered here for the national webinar entitled Scope and Opportunities in Bioscience Research. At the very outset, I would like to welcome on behalf of Rajmata Jijau Mahavidyalaya and also our mother institute, today's resource person, Professor Dr. S. Sir, Department of Botany from Vivekanand Mahavidyalaya, Chhatrapati Sambhajinagar, and another resource person who is also our family member, that means Professor Dr. C.V. Sirsar Sir, who is Head Department of Zoology from MSP Mandals, Vinayak Rao Patil Mahavidyalaya, Vaidapur. Friends, biosciences means botany and zoology department have jointly organized this national webinar, and the theme here, scope and opportunities in biosciences, will definitely help the new researchers, students, and faculty members to update them and to upgrade in various research. Department of Botany Head Dr. Smita Basule, Department of Zoology Head Professor Dr. Ashok Lake Sir have put effort to organize this national event. We know the biosciences research is the study of living things. And as we say that flora and fauna, that means both are connected with all living things, especially our human beings. And its scope and opportunities are vast. But nowadays, as we know that due to hybridization and also due to various human activities, global effect, we know all these biosciences are directly connected with the human beings as they provide us food as well as shelter, as well as medicine, as well as clothes. So we know the contamination in water have resulted number of species where they are diminishing, and also in plant kingdom also number of species of plants, which we say that the country plants are diminished, and hybrid plants which have less immunity, or you may say that their life is very less, they cannot survive in drought conditions. Such challenges are there in today's life for the human beings and also living beings. We also know that the animals, or we may say mostly cattle, which feed us by giving milk, which is the important ingredient of human being. It is also contaminated due to these hybrid varieties and we can say that they directly affect to the human being by causing various abnormalities, various diseases to the human beings. And the scope for this biosciences research for the new budding researchers is that we can switch over towards um, practices which cannot affect the natural uh, process or the natural growth of plants, natural growth of fauna. That maybe we are more dependent upon the marine animals, that means fishes, and most of the world's population is dependent on the marine animals. So to avoid the contamination and to avoid its diminution in the life cycle, we need such subjects to be advanced and to be, we all must be updated for the same. And lastly, feel very warm wishes for this webinar, and request all the participants to connect with this till end. Thank you.
Thank you, sir. Thanks a lot with your presidential address. With a kind permission of both the resource person, now I would like to invite the resource person, today's resource person, Professor Dr. C.V. Sirsat, sir, who is Professor and Head Department of Zoology, Vinayakrao Patil Mahavidyalaya, Vaizapur, for sharing his views on his favorite topic, particularly profile of faunal biodiversity, special reference to birds of India. On this particular topic, Professor Dr. Sirsat sir will enlighten us the profile of faunal biodiversity. Now, I would like to request Professor C.V. Sirsat sir, please. Hello. Hello, Sirsat sir. Hello.
हाय हाँ सर आवाज आ रहा है
येतोय आवाज येतोय तुमचा. Now I would like to request Honorable Professor Dr. C.V. Sirsat sir to share his views on profile of faunal biodiversity, special reference to birds of India. Sirsat sir, please.
Please allow me share my screen.
Kumbare sir.
Hello.
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 सरांना करा म्हणा शेअर करा म्हणा काय पण
Sir, allow करा ते screen.
हो allow
Share
केली सर,
screen.
तुम्ही शेअर करा सर.
तुम्ही शेअर करा. Sirsat
लाके
sir.
सर तुमचा माईक बंद करा लाके सर.
Yes.
वाघमारे सर माईक बंद करा तुमचा.
Screen share होत
पार्टिसिपंटला विनंती आहे स्वतःचे माईक बंद करावेत. पार्टिसिपंट
तिथूनच बंद करा की.
काळे सर
Hello.
All participants are requested.
माईक बंद करा. संजय काळे सर
ते ओपन करून घे.
शेअर होत नाही ना क्रोम ने विंडोज ने.
Right. Full screen.
चालेल. तेव्हा तुम्ही माझ्याकडे बघतो.
हेच नाही ना.
Lakesh sir.
Hello sir.
बोला.
Screen
बोला
दिसते
की.
सर. Hello.
Sound clear.
Screen visible आहे. Hello.
स्क्रीन तुमची शेअर झाली नाही सर अजून.
Hello. एक मिनिट.
शेअर होती सर स्क्रीन शिरसाट सर, स्क्रीन शेअर होते, वेळ लागतो थोडासा.
जादू सर बोलो,
2-4 सेकंद लागतात.
जादू सर बोलो.
हो.
ते वेळेला बरं. एक मिनिट. अरे होत नाही ना, शेअर होत नाही. हॅलो, स्क्रीन शेअरच होत नाही. हॅलो,
स्टॉप शेअरिंग आलं ना. शेअर होतोय ना.
शेअर
शेअर
झाले का सर? शेअर सर होते का बघा तुमच्या मोबाईल?
हॅलो शिरसाट सर.
ऍड केलं पाहिजे आपण.
हा सर,
त्यांना शेअर करा म्हणा.
होत आहे होईल, ते वेळ लागतोय म्हणले.
स्क्रीन शेअर होत नाही सर याच्यात. झाले का? हा.
होते का?
स्टार्टेड स्क्रीन शेअरिंग असा मेसेज आहे, होईल.
हा, युअर स्क्रीन शेअरिंग. पॉज करतो.
प्रिव्ह्यू होत. तेच आहे ना, प्रिव्ह्यू दिसायला पाहिजे ना त्यांना तिथं.
तर दिसतंय का बघा? स्टॉप कसं
एक मिनिट. बरं, विंडोज मध्ये येतं. नॅशनल आता दिसतंय सर, बघा बरं. हा, सराव नाही केलेलं. कॉलेजच चांगलं आहे.
हा झाली सर.
झाली ना?
झाली का?
शिरसाट सर.
ओके सर, ओके.
झालं.
हा सर.
चला, ओके, नाव स्टार्ट युअर.
Am I audible, sir?
Yes, sir.
Hello.
Yes, you are audible, sir.
Okay, sir. Thank you.
Right.
Thank you.
Well,
At the outset, I very much thankful to the organizer of national webinar on scope and opportunities in biosciences research, Principal Dr. Gopal Kakade sir. Also thankful to the convener of this workshop webinar, Dr. Ashok Lakhe sir, organizing secretary Smita madam, and whole team of the webinar. The topic selected by the team, that is the profile of that is the scope and opportunities in biosciences research. Actually, it is the burning issues regarding the biosciences, because biosciences, which includes the specifically the botany, zoology, microbiology, biotechnology, all these are the most important sciences, particularly for the human being, and among which botany and zoology, specifically flora and fauna, as described by the principal Dr. Kakade sir, because these flora and fauna which are interdependent, interrelated to each other, the flora can't live without fauna, and fauna can't live without flora, and therefore it is too much interrelated, and therefore this is the topic which is selected by the organizing committee, which is the most important, because these two branches, these two flora and fauna, which has the most important for the survival of the human being as well as various other uh, animals which are present on this planet Earth. Dear friends, topic for the today's lecture, which is selected by me, that is the profile of faunal biodiversity, special reference to the birds of India. Friends, birds which are the flying quadrupeds, and nowadays that number of these birds, it is decreasing day by day. Not only the birds but the various other animals as well as the plants which are goes down declining, and therefore this topic which is selected by me, profile of faunal biodiversity, special reference to birds of India. I hope this little bit increase your knowledge regarding the flora and fauna, their threats, and various other aspects. Friends, biodiversity, which is nothing but the sum of total flora and fauna which is present on this planet Earth, and that total sum of or total plants and animals, not only the plants and animal but also the microorganisms such as the bacteria, fungi, and various other organisms which are present on this planet Earth. That biodiversity, which is the actually which is the biological diversity, or that is the biosphere, which comprises the complex collection of the innumerable organisms, that is called as the biodiversity. And that world biodiversity, which is described by the Walter in 1986, which includes the protozoans, bacteria, multicellular organisms, plants, fishes, mammals, and various other living organisms which are present on the various ecosystem habitat on the planet Earth. Actually, that biodiversity we can examine at various levels, evolutionary as well as the ecological. All these are the components of the biodiversity, that is the local population, intraspecific, specific, interspecific, macro scale, as well as the on the biosphere level. Friends, the biodiversity which has the multifold values regarding the social values, ecosystem services, economic value, aesthetic value, ethical and moral values, productive use, consumption, environmental value. Friends, particularly in case of the biodiversity, which plays an important value, specifically the process of the uh, purification of the water as well as various other components. And you know very well the water or the pollutant of the or sewage, which is directly poured or carried towards the river, and from the river that contaminated water which is poured into the river, and from the river to the dams which supply to the cities for the drinking purposes, and that water which is purified by the ecosystem, particularly the vegetations which are present in the aquatic ecosystem, various other animals, microorganisms which disintegrate, break down all the waterborne nutrients and pollutants brought into the wetlands, and which plays an important role in the purification of the water. Not only this, that biodiversity, it has an economic value. That biodiversity, various components of the biodiversity, it is a major resource for the various industries, and therefore it has a economic value. Not only this, the aesthetic value, productive use of the consumption, food which obtain, health, as well as the various medicines which we obtain from the natural resources. That is the main important of the biodiversity, and therefore we should have to share for the ethical as well as the moral values of the biodiversity, and we should have to save our biodiversity at each and every step of the life, and therefore we should have to care and save our biodiversity. Each and every step should be the for the saving of the biodiversity is the most need of the time, and I can say that is the burning issue, and therefore topic selected by these opportunities, scope and opportunities in the biosciences, which is selected by the organizing committee of the Rajmata Jijau Mahavidyalaya Dharur, which is the most important nowadays. Friends, biodiversity, which is studied at three levels, that is the diversity, species diversity, diversity of ecosystem, diversity of the genes, and that diversity variability which is observed in case of the species, particularly in case of the species, species diversity of the species, particularly in case of the birds or the avian species. Nowadays in the India, there is near about uh, 1329 species of the birds. That is the diversity of the bird species. Number, it is too much, innumerable number. In case of the ecosystem diversity, that ecosystems which is of various types, that is the forest ecosystem, desert ecosystem, as well as the aquatic ecosystem, it may be the freshwater ecosystem, it may be the marine water, coastal water ecosystem, and so on. Besides this, the genetic diversity, that genetic diversity which is studied at the individual or particular species level, although two individuals of the same species belong within the same species, but it also shows variability within that particular species. Now see, all these are the breeds of the dogs, but genetic constitution of all these dogs, it is variable. Not only this, the genetic variability of the son and father, it is also variable, or it varies individual to individual, and thus genetic biodiversity, or you can say this is the bival. That bival species, individual to individual genetic constitution which is changes, and such a type of the study of the variability in the genetic constitution, which is called as the genetic biodiversity. Now, species biodiversity, these are the horned beetle species of the horned beetles. See the variability in that particular species. Not only this, variability in the donkey, as well as each and every species, it shows the variability, variations in that particular species also. Such type of the species, number of species which of the same population, which is shows the variability, and such type of the species variability, which is called as the variations and variability, which is nothing but the species biodiversity. Ecosystem variability, as earlier described, that is the aquatic ecosystem. It may be the lake, pond, it may be the river ecosystem, it may be the marine water ecosystem, or it may be the forest ecosystem, desert ecosystem, and so on. Such type of the ecosystem variability, ecosystem biodiversity, which is observed on the planet Earth. We should have to concentrate each and every type of the ecosystem, each and every type of the ecosystem, which is nothing but a unit of the environment, and to save our environment, the study of the ecosystem, which is the most important. That ecosystem, which is the self-regulating and self-sufficient and self-regulating system, and if any component, it may be the plant, it may be the any animal, if it is disturbed, total ecosystem, total environment will be disturbed, and therefore the ecosystem biodiversity, study of the ecosystem biodiversity, it is the most important. It may be the any lake, it may be the any pond, it may be the any river. Now the each and every river, it may be the Godavari river, it may be the Mandra river, it may be the Yamuna, it may be the Ganga, all rivers which are polluted, and this pollution which causes the adverse effect on each and every living organism which is present in that ecosystem. Therefore, there is a too much scope, there is too much opportunity for the study of the biodiversity or the ecosystem biodiversity. Now, this is the global species diversity, that is the viruses, protists, fungi, plantae, animalia. These are the species which are described, a total number of species which are described. That is the uh, 15 lakh 10,910, marine species 1,67,000, freshwater 90,000, terrestrial 9 lakh 61,000, symbiotic 31,000, total species, that is the 12 million 62,000 species. Now, specifically in case of the faunal species of the India, protozoan, that is the 3,525. These are the chordates, cephalochordates, urochordates, reptilians, particularly in case of the birds, 1,340, among which the endemic species, which are 6.04%, that is the endemic threatened number of species, that is the 89 threatened species, and this is about the faunal species of the India according to the ZSI 2018. Biodiversity in India, that is the mammals 386, birds 1,290, uh, 1,219, reptiles 495, amphibians 207, freshwater fishes near about 700. These are the data which is of based on the Kumar et al. 2000. These are the various resources for the data collection. Biodiversity in India, that is the molluscan, lichen, various other taxa, angiosperms, bryophyte, lichens, fungi, algae, and so on. Comparative position of the species biodiversity in India. as compared to the mega diversity countries in the world. There are, I like, 12 mega diversities country, and among these higher plants, mammals, birds, reptiles, amphibians, fishes, birds, that is the ninth rank in the, as compared to the mega diversity country. Among 12, the birds constitute the ninth number. Fishes, the estimated number of the fishes which rank first as compared to the mega diversity countries. Mammals at the seventh rank in the mega diversity countries. Now, actually, it is the richness of our country. The fishes stood first, amphibian ninth, birds ninth, mammals seventh, higher plants ninth, as well as the reptiles at the fifth rank. Actually, it is the samruddhi, kiva richness of our country. Why it is richness as compared to the mega diversity countries? Among the mega diversity, South America, North America, Cambodia, India, which is one of the 12th mega diversity country, and among these, the fishes stand first in the mega diversity countries, and large number of the flora and fauna, which is the richness of our countries. But nowadays, that richness of our country, not only the country but of all the countries, it under threat as compared to the past period. There are five mass extinctions. That five mass extinctions in the history nowadays. Hello, hello, hello. आवाज येतो का सर? हॅलो.
येतोय सर, आवाज येतोय. चालू चालू घ्या तुमचं.
And nowadays we are at the sixth mass extinction, and that sixth mass extinction, it is very severe. Why it is severe? Because the rate of extinction of the species किंवा पृथ्वी तलावरून ह्या स्पेसिस नष्ट होण्याचा रेट सध्या जास्तीत जास्त आहे. आणि व्हाय तो जास्त आहे? बिकॉज ऑफ द anthropogenic activities as well as deforestation, pollution, and most important change throughout the world, that is the climate change. And therefore, the sixth mass extinction, which is the severe and very fast, and therefore we should have to take care about uh, the or to save the biodiversity. These mass extinctions of the last five, that is the, which is took near about 443 million year ago, that is at the end of the Ordovician, that is because of the severe ice age led to sea level falling by 100 meters, and that caused the loss of near about 60 to 70 species of the flora and fauna on this planet Earth. Led to next mass extinction, which is took place near about 360 million years at that time, that is the prolonged climate change, and that also killed near about 70%, whatever it means, the species which are present at that time. Among that, 70% species, including all corals, killed in that mass extinction because of prolonged climate change. Next, that is the 250 million years ago, near about 95%, whatever it means, the number of species which are present at that time, that is perished. Next, fourth, that is the 200 million years ago, that is because of the volcanism, that is the volcanic eruption, which caused the loss of large number of species. Fifth one, which took place in the 65 million years ago, and that is the because of the impact of giant asteroid impact, and which lost the dinosaur as well as the amniotic animals, mammals, as well as the number of other species of the fauna also perished, killed from the planet Earth. These are the 5 mass extinctions which are took place on this planet Earth, and now we are at the verge of the sixth mass extinction. And to decrease the rate of this sixth mass extinction, it is the very essential for the zoologist, botanist, as well as the various other scientists to work on this aspect. And according to some workers, that is, the species are becoming extinct at the significantly fast rate on this planet Earth, particularly in case of the birds. The total number of the species of the birds, which is near about 59 to 69, 59 to 60 birds species which have been extinct or lost from the planet Earth. Now this is the data. Nearly half of the 177 mammal species survive, lost more than 80% of their distribution between 1900 to 215. 1900 to 215 च्या दरम्यान म्हणजे 115 वर्षात जवळपास 177 मॅमलच्या स्पेसीस हा पृथ्वी तलावरून नष्ट झालेले आहेत, एक्सटिंक्ट झालेले आहेत. म्हणून हा जो रेट आहे सिक्स्थ मास एक्सटिंक्शनचा, हा जास्त फास्ट आहे. पर इयर जर याचा रेट काढला तर 2 ते 3 स्पेसीस पर इयर हा लॉस होतोय मॅमलचा. मॅमल्स मध्ये ह्युमन बिईंग सुद्धा येतोय, आणि कधी ना कधी आपल्या बऱ्याचशा लोकांचं मत आहे मानव सुद्धा पृथ्वी तलावरून नष्ट होण्याचं शक्यता आहे. त्यामुळे आता जर काळजी घेतली तर माणसाचं आयुष्य पृथ्वी तलावर किंवा अदर ॲनिमल्स आणि प्लांटचं आयुष्य निश्चितपणे वाढेल. त्याची काळजी आजच घेणं गरजेचं आहे. दिस इज द मोस्ट इम्पॉर्टंट लिनियन शॉर्टफॉल किंवा लिनियन शॉर्टफॉल ज्या फ्लोरा अँड फाउना स्टडीड झालेलं आहे ते 1.4 मिलियन झालेलं आहे. त्याच्यातलं फक्त फ्रॅक्शन, पण 5 ते 10 मिलियन स्पेसीज आणखीन अनस्टडीड आहेत, आणखीन त्यांचा अभ्यास झालेला आहे. आणि देअरफॉर देर इज टू मच स्कोप अँड ऑपॉर्च्युनिटी फॉर द फील्ड बायोलॉजिस्ट, इट मीन द झुलॉजिस्ट, इट मीन द बायोलॉजिस्ट. अशा कितीतरी स्पेसीज आहेत त्या आपल्याला अननोन आहेत, आणि त्या अननोनचा शोध घेणं हा यंग जनरेशनचा, यंग बायोलॉजिस्टचा, यंग झुलॉजिस्टचा अतिशय गरजेचं आहे. देर आर दॅट threatened species or the endangered species data which is described by the Sir Peter Scott in 1963. Sir Peter Scott ने a red data book describe केलेला आहे in 1963, and according to that red data book, all the species which are categorized by the IUCN as extinct, endangered, vulnerable, rare, intermediate species. This is the data which is categorized by the IUCN or Sir Peter Scott, unknown, LC that is the less concern, that is the near threatened, VU that is the vulnerable, endangered, critically endangered, extinct in the wild, and EX that is the extinct species. Critical that is the less than 80% in the within the 10 year, or 250 mature individuals, 50 individuals in the very small population. किंवा स्मॉल पॉप्युलेशन असेल तर 250 स्पेसीज असेल तर त्यांना क्रिटिकल समजलं जातं. स्मॉल रेंज मोर दॅन 100 एक्सटेंट ऑफ ऑकरन्स, इट इज मोर दॅन 100 स्क्वेअर किलोमीटर मध्ये जर असेल तर आणि लेस दॅन 80% स्पेसीज जर असतील त्याला क्रिटिकल समजलं जातं. एंडेंजर्ड मध्ये व्हेरी स्मॉल पॉप्युलेशन असेल 250, स्मॉल पॉप्युलेशन असेल 2500 मॅच्युअर इंडिव्हिज्युअल असेल तर एंडेंजर्ड. वल्नरेबल मध्ये दॅट इज द क्रायटेरिया फॉर द व्हर्नेबिलिटी. मोर दॅन 100 मॅच्युअर इंडिव्हिज्युअल असतील स्मॉल पॉप्युलेशन मध्ये तर त्याला व्हर्नेबल स्पेसीज असं कन्सिडर केलं जातं.
Hello. Hello. Hello, sir. Hello. Hello. Shirsat, sir. Hello. Hello. Shirsat, sir.
सर फोन
Hello.
करा सर, एक फोन करा त्यांना.
एक मिनिट हा. Hello. Hello. Shirsat, sir. Hello.
नीटचा
sir.
प्रॉब्लेम. हा येतो
हा
ना
हा
आवाज.
हा. आता सुरू.
हा एक मिनिट. आता स्क्रीन दिसते की सर? हॅलो हॅलो, स्क्रीन दिसते सर? हॅलो,
हा.
विजिबल आहे स्क्रीन?
विजिबल आहे तुमचा.
ओके.
स्क्रीन दिसणार का?
स्लाइड नाही आहे पण असंच चालू करा.
ओके ओके.
दिसतंय सर?
हॅलो,
हा सर.
am I audible sir?
Yes.
Screen visible?
Yes.
Okay. Now this is the data of the India, that is the threatened fauna, that is 648. Among these, according to the Kumar and Khanna, globally threatened Indian fauna, ZSI, that is the 2006. Mammal species 212, ewes 143, reptilia 34, amphibia 148, pisces, molluscan 05, crustaceans 12, other invertebrates 16, total 648 species which are globally threatened in Indian fauna according to the Kumar and Khanna. These are the analysis of the threatened categories: extinct 3, critically endangered, vulnerable, least concern, near threatened, data deficient species. Now these are the birds which are endangered, particularly this uh, white-bellied vulture. It is completely extinct. White-rumped vulture. All these are the critically threatened birds. That is the forest owlet, Himalayan quail, slender-billed vulture, long-billed vulture, white-rumped vulture, and white-bellied vulture. It is totally extinct, not available in the India. Jordan's cruiser, the number it is very less. All these are the critically threatened Indian bird species. Threatened number of the threatened mammals from the India. These are the Asiatic elephant, Asiatic black bear, gaur, wild goat, Himalayan tahr, Nilgiri tahr. All these are the endangered species from the India. Lion-tailed macaco, Nilgiri leaf monkey, leaf-nosed bat, slow loris, Indian rhinoceros, Asiatic wild ass. All these are the critically endangered species. Endangered species at present approximately that is 81 species of the mammals, 31 species, 38 species of the birds, and 18 species of the amphibian and reptiles, which are nowadays endangered. Critically endangered mammals. These are the pig, flying squirrel, Salim Salif fruit bat, free-tailed bat, which are endangered, pygmy hawk, Malabar horse spotted civet cat, that is the Sumatran rhinoceros, which are endangered mammals. These are the critically threatened, that is the free-tailed bat, fruit bat, Malabar civet cat, pygmy hawk, which are the endangered mammals. That is the Barasinga, four-horned antelope, black buck, Ganges river dolphin, great Indian bustard, which is very endangered, particularly in case of that great Indian bustard, near about uh, 50 years back in the Marathwada, there is presence of large number of these uh, great Indian bustard. But nowadays there is no any single bustard in the Marathwada except few in the Solapur district. Python, Trishu, which are the endangered species. These are the factors which are affecting the loss of the decrease in the number of the species, flora and fauna. That is the habitat loss in the ocean, habitat loss on the land, habitat loss in fresh water, pollution, introduced species, over harvesting, global climate change, uh, secondary extinction, that is one species becomes extinct, there will be the many other extinctions as the consequences: deforestation, desertification, urbanization, wetland drainage, pollution, dams and water diversions. All these are the factors which are affecting the extinction and uh, causing the loss of the species. Therefore, save biodiversity and save life on the, it means the flora and fauna which is present on this planet Earth. And therefore, each and every one should have to share their own for the saving of the biodiversity and life on this planet Earth, because each and every species which is most important in the food chain as well as the food web and for the cycling of the materials. These are the conservation methods, in situ, ex situ methods for the conservation of the birds. Uh, India, which is richest in the flora and fauna among the 12 countries, India, which is one of the country among the 12 mega diversity as described earlier. India, Brazil, Colombia, Ecuador, Peru, Mexico, Madagascar, Zaire, Australia, China, Indonesia, and Malaysia are the mega diversity countries. These are the hotspot, 25 hotspot, hotspot throughout the world, out of which the two are in India, that is the Eastern Himalaya and Western Ghats. There are 34 terrestrial hotspots in the world, in India, Eastern Himalaya and Western Ghats. These are the various hotspots throughout the world. The 23 heritage sites in the India. There are 14 existing biosphere reserves in the India. These are the 14 biosphere reserves. Ramsar sites 25. That is aquatic bodies. In India, for the conservation, 95 national parks, 500 wildlife sanctuaries, and 2 conservation reserves for the saving as well as conservation of the flora and fauna. Tiger reserves in the India, that is the 9, and nowadays that is the 28. These are the status of the total diversity of the Indian fauna, endemic species, marine biodiversity of India, biodiversity of the India, major hotspot of endemic and genetic diversity of India. Species endemism among the plants, that is the 33%. Endemism among the animals, that is the mammals, which are 44 species, birds 55, reptiles 187, amphibia 110. Therefore, we should have to care and save our biodiversity. What can we do for the save and caring of the biodiversity? That is the spend time outdoors, plant trees, support companies that work in ways to reduce environmental damage, learn more about the biodiversity. If we save our biodiversity, we can save ourselves. Major threats to the biodiversity: limited distribution. Now this is myself, the survey of the avian biodiversity from the various aquatic bodies, freshwater bodies. This is the richness of the Marathwada, different birds from the different areas of the Marathwada. This is the little bit support and saving of the biodiversity. Hello, hello, hello. Sir, आवाज येतो का?
येतो.
हां.
Yes, sir. Yes, you are audible.
This is the little bit share for the conservation of the biodiversity. We support such type of the events and save the various components, snakes, birds from the our locality. These are the poisonous as well as the non-poisonous snakes, which are saved from our area at different times or from time to time. Or that is the rescue operation for the snakes as well as the birds by myself. This is the poisonous cobra which is collected from this well. This is the cobra which is collected visible. दिसतंय का हे? Hello, hello. Screen दिसते का sir? This is my trial for the saving of the birds from the area. Egret, Maina. This is the shelduck, crow, and various other birds, bulbul, and so on. This is my service for the birds, different birds, time to time. These are the share. This is the uh, black shouldered kite, which is saved by me and my companies. This is the little bit service for the life of the birds and various other animals. Myself. This is the survey of birds from the Marathwada. Various types of the birds.
sir. Hello. Hello. Hello, Shirsat sir.
View option, view option.
Hello, sir. Now, I am very much thankful to Honorable Professor Dr. C.V. Sirsat for his valuable and informative speech. On our request, sir, you have spent your valuable time for us. So I am again very much thankful to you. Thanks again. So then I would like to request Honorable Smita Basale ma'am to introduce the another resource person, Professor Dr. A.S. Bukhtar sir, Professor and Head, Department of Botany, Vivekananda College, Chhatrapati Sambhajinagar. Please, Basale ma'am.
Yes, sir. Thank you.
Yes.
Am I audible and visible?
Yes, yes.
Okay, good. Thank you very much, sir, for the Shirsat sir for giving us such a valuable talk on the biodiversity, importance of biodiversity, and how to take care of biodiversity to survive. Now, here we are having with us our second speaker, Professor Anil Bhuktar sir. He is head of Department of Botany, Vivekanand College, Chhatrapati Sambhajinagar. He completed his education with doctorate degree in 1995. Since 1995, he is engaging himself in the research field. He has 33 years of experience in teaching and research field. He continued his research till today. He guided 19 research students to obtain their doctorate degree. A huge number of papers, that is 123 papers, are published by him in various national and international journals. He also wrote 2 textbooks for the BSc student according to our new education policy. He guided 19 students. His area of research interest is taxonomy, pharmacognosy, and phytochemistry. He was also honored by the award Vivekanand Shikshan Shri. He successfully shouldered the responsibility of in-charge principal in last few years. Now, may I request to Bhuktar sir to please deliver your valuable lecture. Over to Bhuktar sir.
Okay. As I outset, very much thankful to organizer who have given me an opportunity to deliver my lecture. I am very much thankful to principal of Jijamata Mahavidyalaya
Rajmata.
Dharu, Rajmata
Rajmata.
Mahavidyalaya
Jizau.
Dharu. Then convener Dr. Lakde sir, organizing
Lake.
secretary Smita Basole, and management. My topic of research or to deliver here, that is phytochemistry of medicinal plants. So phytochemistry means the chemicals, that is the secondary metabolites which are present in the plant, and medicinal plant means the plants which cure our diseases, disorders, and ailment. For that purpose, we use our diffusion, then kadhas, or whole plants, and different parts of the medicinal plants. So phytochemistry of medicinal plants is essential for identifying and analyzing the bioactive compound that contribute to their therapeutic properties. By studying the chemical profile of plants, especially tubers, corms, and rhizomes, research can be isolated important compounds like alkaloid, flavonoids, and glycosides that are responsible for the healing effect. Tuber, corms, and rhizomes often store secondary metabolites in high concentration, making them a rich source of potent medicinal compound. Thus, understanding is crucial for the development of new drugs, improving the efficacy of herbal remedies, and ensure quality control in the production of plant-based medicine. High resolution liquid chromatography and mass spectroscopy, that is HRL CMS, is a powerful analytical tool used in the phytochemical analysis of medicinal plants. It combines the separation capabilities of liquid chromatography, that is LC, with the precise mass detection of mass spectroscopy, that is mass MS, allowing researchers to identify and qualify a wide range of bioactive compounds in complex plant extract. HRLCMS is particularly useful for profiling metabolites even in trace amount, and can differentiate between structurally similar compounds. This technology play a vital role in understanding the chemical composition of medicinal plants, ensuring the consistency of natural remedies and facilitating a discovery of new therapeutic agent. For that purpose, we have used materials and methods. First of all, we have collected specimens were collected from the different region of Maharashtra state. The exit data of the plant species are given along with the date of collection and field numbers. The detailed morphological characters were studied using floristic literature like Flora of Cook, Dr. Naik, Sharma, S.R. Yadav, and Sardesai. The herbarium were prepared and deposited in Vivekanand Herbarium, that is VH Herbarium, which is affiliated to New York Botanical Garden, America. The Department of Botany, Vivekanand Art, Sardar Dilip Singh Commerce and Science College, Aurangabad, or now it is known as Chhatrapati Sambhajinagar. The specimens were compared critically with the authorized herbarium specimen in the herbaria, that is BSI Pune, Shivaji University Kolhapur, and Dr. Babasaheb Ambedkar Marathwada University Chhatrapati Sambhajinagar, Aurangabad. The combs, tuber, different parts of the plants were set dried and chopped into small pieces. Then the fine powder was made with a mortar and pestle. Crude plant extract was prepared by the Shockless extraction method. The extract was sent to a sophisticated analytical instrument facility, SAPE, IIT Powai, Mumbai, for RCLCMS. The powder was stored in airtight container for further phytochemical studies. So these are the procedure, that is washing of the material, weighing of the material, that is drying, then sock slate equipment, then sample is loaded in sock slate.
Being recorded.
That is extracted and proceed for the further phytochemical analysis to HR, LCMS techniques. Modern medicine, that is the importance of and analysis in the modern medicine. The medicinal plants are of, as already I have told, are of great importance to the health of individual and communities. The medicinal value of these plants lies in some chemical substances that produce a definite physiological action on a human body. The most important of these bioactive constituents of plants are alkaloid, stannine, flavonoid, and phenolic compound. Many of these indigenous medicinal plants are used as a spices and food plants. So I have selected 5 plants to deliver here. The first plant is, that is Gingiber officinalis. Second is Curcuma ambada. Then Solanum melongia. Then Amorophilus folius and Dioscorea tomentosa. So the vernacular name of Gingiber officinalis is adrak, sukku, in Telugu almu, then Malayalam inji, then Sanskrit adrak, adrika, and English ginger, and several names are there. The description of the plant is, this is the perennial rhizomous herb. Rhizome yellowish inside, thick, palmately lobed, pungent in taste. Leaf shoot is 1 to 1.5 centimeter long. Leaves sessile, ligulate, ligule membranous. Leaf base hairy, lamina narrow, lanceolate, 20 to 35 by 1.5 to 2 centimeter. Upper surface is glabrous and lower is hairy. Inflorescence is 15 to 25 centimeter long lipless peduncle. A spike is 3 to 6 into 2 to 3 centimeter long, oid, bracts, green, 2.6 to 1.8 centimeter, turning to red at maturity. Bracteoles almost equal to bracts. Ovate, white, membranous, 2.5 to 1.9 centimeter. Flowers longer than bract, fragile, calyx tubular, 1 to 1.3 centimeter, unilaterally split, corolla tubular, pale yellow, lobes almost equal, lateral lobes 2 to 2.5 centimeter, labellum dark purple with creamy yellow blot under thicky parallel connective yellow, crease dark purple, stigma white, margin ciliate, prolonged above, epigynous gland free from each other, ovary tricarpellary, trilocular with axial placentation, seed not collected or not found, and a flowering and fruiting time is August to October, and we have collected that is from Aurangabad. The medicinal uses of Gingiber officinalis. Gingiber officinalis is the best known gingiber plant in Gingiberi family, and it is also referred to a garden ginger or common ginger. This ginger is used in Ayurveda both fresh and dried preparation for curing heart problems, treating stomach ache, upset, diarrhea, headache, and nausea. Other than the Gingiber officinalis, has also been used to promote the release of bile from the gallbladder, then reduce joint pain from arthritis, treat heart and lung diseases, relieve cough and cold, throat infection, and even the removal of worm and comb. In the Chinese and Japanese system of medicine, fresh ginger is used for its warming properties and as a remedy for cough and nausea, whereas dried ginger is indicated for alignment of the digestive system. So the Gingiber officinalis plant has the properties like antimicrobial activity, anti-diabetic activity, anti-cancer activity, anti-inflammatory activity, anti-pyretic activity, immunomodulatory activity, anti-obesity activity, anti-platelet aggregation activity, larvicidal activity, neuroprotective activity, and a cardiovascular activity. Due to this reason, we have selected this plant. The activity of the phytochemical compounds identified in major abundant metabolites from Gingiber officinalis. This is the Gingiber officinalis plant. Here the inflorescence is there and the rhizome is there. So as I have told, we have sent the material for the HRCML-CMLs in various peaks are obtained from the chemicals constituent. So in the chart, the peak height, then a retention time, and peak numbers are mentioned. So on the basis of this peak, we can find out the its group, chemical group, which are associated, then mass spectra like choline, then salicine, dimethylglycine, oxyperonol, dihydrotetrabenzine, toluene, likewise, and this is the retention time. This is the mass spectra, And this is the functional group, that is chemical substances which are associated with that chemical structure. So these are several chemical structure which are found. The next plant which we have selected, that is the turmeric. The both plants are belonging to family Gingiberaceae. The turmeric, its botanical name is Curcuma ambara. The plant which is used in our daily, that is the Curcuma longa. We have selected Curcuma ambara, or it is commonly known as Ambi haldi, Amba haldi, then Amba halad, Amba haldi in different local languages. The morphology of the plant is herb, leafy shoot 2 to 3 feet or more, rhizome large, yellow inside, having smell of green mango. The tubers are cylindrical, branched, horizontal. The root tubers is absent. Leaves are 4 to 6, large, hairy or glabrous along the lanceolate, acute, narrowed at the base, and 40 to 65 by 12 to 15 centimeter, petiole as long as lamina. The inflorescence is central, peduncle, 10 to 15 centimeter or more. Spike slit are 7.5 to 12.5 centimeter long. Chromobract hues at the base, light green, 6 to 8. Fertile bract, 15 to 20, orbicular, absute, and slightly recur. Flowers as long as bract, calyx 3-lobed at the tip, cleft, one side, 1 centimeter long, corolla whitish pale yellow, corolla tube, funnel shape, 2.5 to 3 centimeter long, labrum, 3-lobed, pale yellow with dark yellow bands, lateral, staminoid pale yellow, slightly recurved at apex, stigma appressed within another lobe, style long, filiform, orid, trigonous, tricarpellary, syncarpous, ovules many, 3 millimeter long, fruit is not seen. We have collected this plant from Buldana and Konkan area. The flowering season is June to September, and exicata from Khamgaon, particularly Buldana districts. The medicinal uses of this plant, that is Curcuma ambara, that is anti-tubercular activity, then hypoglycemic, anti-hyperglycemic activity, anti-bacterial and antifungal activity is there, then antioxidant activity and anti-inflammatory, anti-allergenic, hypotriglyceremic activity, CNS depression and analgesic activity on this plant is used. So this is the Curcuma ambara. This is the habitat, and these are the inflorescence. So again, the Curcuma ambara shows there are peaks. There are many types, and the chemical compound which is isolated with the help of that peaks, that is Lambda 23, methylphenyl, 1-methylene, then apomorphine, 3-beta, then elephantine, derugobin, OS spheroid. So on the basis of this chemical structure, which we have found, that may be a new drugs. So next plant is Solanum melongia. That is a vernacular name is, in English it is known as brinjal, or commonly known as eggplant. In Hindi it is known as baingan. In Marathi it is known as wangi. And different languages they are different name. The plant is an annual and erect or suppressant herbaceous up to 50 to 60 centimeter tall, unarmed, but sometimes armed, pubescent with stellate hair. Leaves are simple, subopposite, large, entire blade, ovate or oblong ovate, 6 to 20 by 5 to 10 centimeter long, lobed, base oblique, margin sinuate, lobes, apex upshoot, petiole 2 to 4 centimeter long. Inflorescence mostly racemose, peduncle, obsolete, flower, andromonaceous, pedicel 1 to 2 centimeter long, calyx stellate, tomentose, often with 3 millimeter prickles, abaxillary, lobe lanceolate, corolla, purple or whitish, rotate or lobe dotted, stamen 5 to 8 to 9 millimeter long, anther lanceolate 6 to 7 millimeter long, yellow, filaments 2.5 millimeter long, style 4 to 7 millimeter, fruit large, berry, drupe, white, black, purple, pink, brown, yellow, yellowish, or sometimes green when completely mature, greatly variable in form of size, mostly more than 6 centimeter in diameter, fruit capped with thick persistent calyx, seed many, yellow discoid. This plant is distributed in Maharashtra, that is cultivated as a vegetable in all over the India as well as Maharashtra state, flowering and fruiting June to December, and we have collected this plant from Jalgaon. So these are the medicinal and other uses. The roots are laxative, analgesic, cardiotonic, used in inflammation, neuralgic, and cardiac, debility and ulcer in nose. The leaves are used salagogue, narcotic, antihypetic, used in cholera, bronchitis, asthma, odontalgia, and fever. So this is the plant. Because of its white fruits, it is known as eggplant. Several varieties are there. This is the HRAMC chromatogram of Solanum melongia. The peak shows different rotation time. These are the chemical substances, that is retention time, mass, and formula. Next. Another plant which is we have selected that are known as elephant foot yam. That is, botanical name is Amarophophilus poenifolius. The vernacular name of this plant in Sanskrit, it is known as Suran. In English, it is known as foot yam. In Marathi, it is also known as Suran, or in Hindi, it is known as Surankan. And in different languages, the vernacular name is given. This is the tuberous herb, tuber rub, depressed bulbous, 8 to 15 into 10 to 18 centimeter, leaf solitary, petiolate, petiole smooth, rarely slightly rub at the basal half, 75 to 150 centimeter long, 6 to 10 centimeter in diameter, green with ovide and elongated ovide pale green blotches at and minute pale green spot in between. Extreme base is white, leafless are sessile, large, 15 to 25 centimeter long, inflorescence peduncle short, smooth, whitish yellow, peduncle as a petiolite, 3 to 4 centimeter long, 1.5 centimeter in diameter, in flowering stage, Cataphyllus, pinkish white, colored with irregular brooches. Spathe margin undulate, tip acute, convolvulate portion green yellow, expanded portion is purplish green, with blotches dark purple outside, whitish blotches are observed. And the inflorescence is paddix and sessile. Female flower zone and male flower zone is also present in spadic, and spadic is appendix hemispherical. So flowers and fruiting found in May and November. Its distribution is Vaibhavwadi Taluka, Sindhudurg district, Kolhapur and Pune. The medicinal and other uses: appetite, gastric stimulant, digestive and anti-helmintic. Beneficial in dyspepsia, cough, vomiting, piles, colic and abdominal lump. Petiolol, fermented juice, anti-diarrheal. Calm, anti-asthmatic, anti-dysenteric, anti-emetic, carminative, expectorant, restorative, stomach ache and tonic. In diaspia, enlargement of spleen, elephantiasis, hemorrhoids, abdominal pain, piles and tumors. Seeds and corms are also used as a local application in rheumatic swellings. Root, emagogue, useful in boils and ophthalmia. So this is the habitat of the plant, that is the Amorphophilus poinetiums. So this is the vegetative structure. These are the inflorescence, and this long portion consists of seed that is immature now. So this is the chromatogram of this sample. We have found several peaks, and the peaks, that is marmimal, Chiariosporidol, nicophytine, glutamine, sulfophinin, and these are the chemical substances, these are the retention time, these are the mass spectra, and these are the chemical formula. Another plant that is its vernacular name is thorny yam, Kantelo in Marathi, Kantela alu, and several languages. The leaves are 3 to 5 foliate, rarely unifoliate, mixed leaflet overhead, thick 8 to 20 into 3 to 10 centimeter, cunate at base, acuminate or cuspidate. Lateral often very oblique, dark green and pubescent. On the upper surface, densely white, tomentose beneath, shortly petiolate. Common petioles 3 to 6 centimeter long, male flowers 1 to 3 centimeter long, spike arranged in slender tomentose panicles, perianth green white 1.5 to 2 millimeter long, segment ovate, oblong, acute, subequal, stamens 3, staminoids 3, as long as the perianth. Female flower is in axillary 1 to 4 net or 15 to 20 centimeter long spike. Perianth as in male, capsule, cunate, oblong, 3 wing, downy, retus at both ends, seed 8 to 15 millimeter long, including the wings. It is found in Aurangabad, Bid and Nanded district, and the flowering season is and fruiting season is July to October. The medicinal uses of this plant, that is medicinal properties, anti-inflammatory is there, antioxidant is there, antimicrobial is there, antiplasmotic is there, diuretic there, imagoguing, regulate the maternal flow, expectorant and tonic for general health and vitality. So digestive issue is diarrhea, dysentery, dyspepsia and indigenous. Respiratory problem, that is bronchitis, cough and asthma. Maternal disorder, that is dysmenorrhea, amenorrhea and menomagnegia. Skin condition, eczema, acne and dermatitis. Fever and rheumatism. Then urinary infection and anxiety and stress. So this is the morphology of Dioscorea tomentosa. Basically, there are male and female flowers are plants are separate. So this is habitat. This is male flower. This female flower, female inflorescence is there. Then figure D is a fruit and figure E is a tuber. So these are the chromatogram and the chemical substances which we have obtained, that is neurominic acid, then acetyphenyl, beta naphthalene, ester, dimethyl, laminothyl, respiratory 14 alpha hydroxyphylline, limacillin, oleosilin, ethanolamide, terminaline, and several. The retention time, mass spectrum. Then conclusion, we have what may be the 5 plant presented here. the phytochemical analysis reveals diverse bioactive compound in ginger, turmeric, solanum, Amorphophilus, and Dioscorea, demonstrating potential medicinal properties. The result underscore the significance of traditional medicine and the importance of conservation of sustainable utilization of these plant species. Further research is want to fully explore their therapeutic potential and the development of novel phytopharmaceuticals. These plants offer promising solution for preventing a curative healthcare along with the back to the nature approach. Further research is necessary to underlog their therapeutic potential and development novel phytochemicals. Thank you very much.
Thank you, sir.
Dr. Anil. Thank you very much for your priceless information. Definitely to overcome with side effect of allopathy, some of our plants and Ayurveda treatment help us to the cure some serious diseases. Over to Lakhi sir.
Okay, thank you, madam. Now, I would like to request Honorable Dr. L.B. Jadhwar, sir, for vote of thanks. Jadhwar, sir.
Okay, okay. Hello, sir.
Yes, sir. Yes.
Yes, yes. And are you
Yes,
getting me,
continue.
sir?
Yes.
Okay, thank you,
Yes,
sir.
you are audible. Yes.
Friends, I am here to express my vote of thanks on behalf of the Department of Botany, Department of Zoology, and IQAC of the MSP Mandals, Rajmata Jijao Mahavidyalaya Kilidharu. First of all, I am thankful to the college principal, Honorable Dr. G.K. Kakade sir, for giving his valuable presentee and guiding and supporting to us in organizing the national webinar on scope and opportunities in bioscience and research. Jointly organized by the Department of Botany and Zoology and IQAC of this college. I express my sincere gratitudes to Professor Dr. C.V. Sirsat sir, HOD of Zoology Department, Vinayakrao Patil Mahavidyalaya, Vaizapur, for giving his excellent talk on the title Profile of Fungal Biodiversity, special reference to birds of India. He focused on the burning issue of ecosystem. His talk was very excellent and informative. Certainly, our students be aware for the ecosystem. Again, he appealed to our students to save our environments and ecosystem. Certainly, our students will give a positive response to save the environment. Again, he gave a message, if you save the biodiversity, then we will save ourselves. This message is certainly important to our society. Again, I am thankful to the another chief guest, Honorable Professor Dr. A.S. Buktar sir, HOD of Botany Department, Vivekanand College, Chhatrapati Sambhaji Nagar, for giving his talk on the title Phytochemistry of Medical Plants. His talk was again excellent and informative. His teaching style is very excellent. Again, his way of representation is also very best and will be followed. I express my sincere thanks for giving his kind presenting and delivering a nice talk. I am also thankful to my dear colleagues, vice principal and students for giving their positive response and giving their best presenting to this event. Thank you once again on behalf of this college, and by the permission of the principal, I declare that this event is over. Thank you to the organizing committee for giving me a chance to express my vote of thanks. Thank you, thank you.
राकेश सर करू का यांनी मीटिंग?